Bismillahirrahmanirrahim. We are going to discuss some basics related to the respiratory pathology. Basics for respiratory pathology. Okay. So in this basics, we are going to cover the respiratory tract and the corresponding histology of those respiratory tract. Okay. So mainly we are going to concentrate on trachea, uh, bronchus, bronchioles, alveoli, and alveolar capillaries. So these are the areas we are going to mainly discuss on in respective to histology. Okay. So you all know the respiratory tract starts from the nasal cavity, then the naso oro laryngopharynx comes, then larynx comes. Okay, then they enter the trachea, then trachea gives main branches of bronchi, then the bronchi leads to bronchioles, and they end in the alveoli. Okay, so this is the respiratory tract. So let us draw from trachea. This is trachea, and trachea gives two main branches, that is called bronchi, left and right bronchus. Then this area is called, there is some projection from anterior to posterior, it goes septum like projection. This is called carina, very important area. Inshallah I'll discuss while discussing tumors. Carina. Okay, now this bronchi enters the lung. Okay, they enter the lung, and if it is a right lung, they have three lobes, so they give three branches. Okay, then they give small branches, so we call them as primary, secondary, tertiary bronchioles. They give more branches. Okay, these branches are called bronchioles. So bronchioles, we have primary, secondary, tertiary, and respiratory bronchioles. Okay, so they all end up in the they form alveoli. Okay, they form alveoli. Like that. Then from the heart, from the right ventricle, the pulmonary artery brings the deoxygenated blood. Pulmonary artery brings the deoxygenated blood. This is right atrium, right ventricle. Okay. Okay, pulmonary artery enters the lung, and they gives branches. They become small arterioles, and they become arteries, small capillaries, and the capillaries winds the surrounds the all the glomeruli. The capillary surrounds all the glomeruli. Okay. So like that, the capillary comes from the pulmonary artery, surrounds all the glomeruli, and they come out as a venules. Okay, they come out as a venules, and all the venules join and they form pulmonary vein. This pulmonary vein goes to the left atrium. This pulmonary vein goes to the left atrium. So this is left atrium, left ventricle. Okay, so this is how they are arranged, and this is the alveoli, and this is the pulmonary capillaries, and the area between the alveoli and the pulmonary capillaries, this area, okay, these areas are called interstitium. These areas are called interstitium, pulmonary interstitium. Okay, now you all know the lung is covered by the pleura, covered by the pleura. Remember, the pleura has two layers. One layer is closely attached with the lung. It is literally stick with the lung. Okay, and in between this two layer we have fluids. We have fluids that is called pleural fluid. That is called pleural fluid. This is pleura. This is pleura. Okay, now this is the structures related to the lower respiratory tract, and now let us discuss histology of these structures. Now what I am going to do is I am going to cut the trachea. If I cut the trachea, okay, now I made a cut here, and if then the trachea looks like a pipe. Trachea looks like a pipe, right? In the pipe you can see the holes of the pipe and the pipe wall. You can see the pipe wall. Okay, so pipe has a inside the hole where the air goes inside, and they also have a pipe wall. So I'm going to show you in microscopy this structure. Okay, so you have a trachea, and this is the inside pipe opening. It is called lumen. It is called lumen, and this lumen is lined by ciliated pseudo stratified columnar epithelium. Ciliated pseudo stratified columnar epithelium. This epithelium sits on the basement membrane. That basement membrane is very thick in trachea. It is very thick in trachea. The basement membrane is very thick in trachea. Okay, I told it is a ciliated pseudostratified columnar epithelium, right? And you are seeing cilia. Cilia are the small finger-like projections. It's seen in the epithelium. The cilia is very important because the cilia always moves. It moves in an outward direction, so it sends the bacteria, anything which enters or to outside. Okay, that will not allow anything to come inside. So the cilia will always keep on moving in a outward direction. Okay, now this is ciliated pseudostratified columnar epithelium. Remember, cilia is very important. The cilia is very important. So in the smokers the cilia is affected, and there is another syndrome called Kartinger syndrome where the cilia becomes immotile. Okay, so now let's discuss the histology. So the cell sits on the basement membrane, and the basement membrane is thick in the trachea. Now below that basement membrane we have very thin connective tissues. We have very thin connective tissues. Okay, this has thin connective tissue, and they also have small blood vessels, small tiny blood vessels. Okay, so this is called lamina propria. This area is called lamina propria. Okay, now let's mark the areas. So this is ciliated, pseudo stratified columnar epithelium. Okay, then this is the basement membrane. Then this small connective tissue and blood vessels are called lamina propria. Okay, below that we have very dense connective tissue. Okay, this whole structure are called mucosa. This whole structure are called mucosa. Called mucosa. If you want, I will write it clearly. Written. This whole structure are called mucosa. Okay, now below this, okay, we have dense connective tissue. We have dense connective tissue. We have dense connective tissue, and this dense connective tissue has very dense collagen, and they also have some blood vessels, large blood vessels, and below that we have some glands. They secrete thin fluid. We have some glands that secrete thin fluid. This glands has some pipe that is called ducts that connects and to the mucosa, and this thin fluid is coats the lumen. It coats the lumen. Very thin fluid. It coats the lumen. It lubricates the lumen. Okay, so they have glands. Okay, so these glands are just below the mucosa, so they are called submucosal glands. Okay, then they have cartilage. Trachea has cartilage. Trachea has cartilage and some bone formation. Bone formation also seen. Okay, trachea has cartilage. So this is cartilage and some area they have bone formation. So this is all about trachea. So once again, this is the area below the lamina propria is called submucosa. So in the submucosa we have submucosal glands, and after that we have cartilage and bone. So this is the characteristic feature of trachea. Okay, histological feature of trachea. Now let's see the bronchus. The bronchus is also a pipe, and inside they have lumen, and lumen is lined by ciliated pseudo stratified columnar epithelium. Lumen is also lined by ciliated pseudo stratified columnar epithelium. Okay, and they also have nucleus. They are lined by ciliated pseudo stratified columnar epithelium, actually which sits on the basement membrane. In bronchus, the basement membrane is not very thick like trachea. So the basement membrane is thin. The basement membrane is thin. The basement membrane is thin. Okay, and below the basement membrane we have small loose tissues. Loose tissues and tiny blood vessels. They are lamina propria. And remember, they also have slight elastic tissue. Yeah, elastic tissue. Okay, they also have elastic tissue. So this whole area is called mucosa. Just below that we have in the muscle layer, which is not seen in the trachea, it is seen in the bronchus. So this muscle layer is characteristically seen in the bronchus. These are the circular muscle layers. These layers are called muscularis. These layers are called muscularis. So we have this full area is called mucosa, and this muscle layer is called muscularis, and then they have glands. Glands. These glands secrete mucus. So this is below the mucosa. It is called submucosal glands. So this gland secrete, and the ducts takes the secretion to the lumen, and they will be coated. It lubricates the lumen. Okay, so then we have cartilage. Okay, then we have cartilage. Okay, so this is the structure of this is the structure of bronchus. Okay, so what is the difference between trachea and bronchus? Trachea, they don't have muscularis, so bronchi has muscle layer, and other structures are all similar to that of the trachea. So this is a histology of bronchus. Remember why? That's why in the bronchus, the muscle, when since the muscle is there, the bronchi, if the muscle contracts, the bronchi, for example, if the muscle contracts, what happens? The bronchi go for constriction. The lumen will become constrict when the muscle contracts. When the muscle contracts, when the muscle is contracting, okay, then the bronchi goes for constriction. If the muscle is relaxing, this is broncho constriction. If the muscle is relaxing, the bronchial muscles are relaxing, then the bronchi will go for the bronchi muscles are relaxing, the bronchi will go for bronchodilation. Go dilation. So we give certain drugs to go for bronchodilation in the body during inflammation. Certain chemical substances can cause bronchoconstriction. Okay, if there is bronchoconstriction, the airway is obstructed. Okay, so these are the importance of knowing the histology. Okay, so in trachea we don't have muscle, in the bronchi we have muscle, and the rest of the structures are similar with the trachea and the bone. Sorry, trachea and the bronchus. Now let us discuss about bronchioles. If you see the bronchioles also has a lumen and they are lined by ciliated pseudostratified columnar epithelium. Okay, and they also have basement membrane, thin basement membrane. They have thin basement membrane, and they also have a thin connective tissue. Okay, and small blood vessels, that is called lamina propria. Okay, they have small blood vessels. Below that they have a muscle, muscularis, circular muscle fibers. Okay, they don't have no glands or cartilage in bronchiole. So the bronchioles, they don't have secretion, they don't have cartilage. So because there is no glands and cartilage. So what happens? Because of muscle, the bronchiolar constriction can happen, bronchiolar dilation can happen. Okay. So one more thing I want to discuss in the mucosa of the bronchioles. Let us draw the mucosa epithelium of the bronchioles in detail. So this is the basement membrane of the bronchioles, and we have epithelial cells, ciliated pseudostratified columnar epithelial cells. In between them, there are special cells. They have granules. Granules. They have These are the special cells. They have granules, and there are another group of cells. This is normal ciliated cells. And another group of cells. They have granules at the base. They have granules at the base. The granules at the top near to the lumen, these cells are called Clara cells. These Clara cells secrete CC6. Okay, the CC6 coats the bronchiole So when you are expelling the air, the bronchioles will not attach together. So bronchiole is not a rigid pipe because they don't have cartilage. They are very smooth pipe. When the air is expelled out, then the bronchiole can attach. The lumen of the bronchioles can attach, and to avoid that, we have a CC6 which coats the bronchi. Okay, so this is comes from the Clara cells. If bronchioles are damaged, the CC6 can be seen in the sputum, or the CC6 can go to the alveoli and it can diffuse into the blood, and you can detect in the blood CC6. So this is the importance of knowing the CC6. Okay, now after the bronchiole, it goes to the alveoli. So now let's draw the alveoli. This is the alveoli. If you cut, see for example, this is the alveoli, and if you cut the alveoli like this, what you will see? You see a alveoli, right? From above, if you see, alveoli will be like this. These alveoli are lined by two types of epithelium. One is flat, one is other one is cuboidal. Okay, the flat epithelium are called type 1 alveolar epithelium. So this cuboidal epithelium is called type 2 alveolar epithelium. So otherwise called as type 2 pneumocytes. Otherwise called as type 1 pneumocytes. Okay, this type 2 pneumocytes secrete surfactant. They secrete surfactant. Okay, it is otherwise called as dipalmityl phosphatidylcholine. Very important because they increase the surface area of the lung. They increase the surface area of the lung. Okay, so these are the epithelium. These are the lining epithelium of the alveoli. So let us draw alveoli in a different. This is the alveoli, and this is the bronchiole lined by ciliated pseudostratified columnar epithelium. Then the alveoli enter the alveoli, they become flat as a type 1 epithelium, and some cells are cuboidal, they are called type 2 epithelium. Flat cells are called type 1 epithelium, and cuboidal cells are called type 2 epithelium, and type 1, type 1, type 1, this is type 2, type 1, and this is ciliated pseudo stratified columnar epithelium. Okay, so now all they have nucleus. They have nucleus. Okay, all the cells has nucleus. Okay, okay. Now inside the alveoli we have macrophage, that is called alveolar macrophage. Alveolar macrophage, we have macrophage. Now you all know the capillaries surrounds the alveoli. The capillary surrounds the alveoli. Now capillaries are lined by endothelial cells. The capillaries are lined by endothelial cells. Okay, capillaries are lined by endothelial cells. Okay, so this is the area of gas exchange. Okay, so what is the area of gas exchange? Alveolar epithelium and alveolar capillaries, that is pulmonary capillary endothelium, and the see, this epithelium sits on the membrane, right? So we call it as a alveolar epithelial membrane that contains alveolar epithelium, and the capillaries, we call it as a alveolar capillary membrane that contains endothelium. In between them we have a interstitium. See, now interstitium is normally not even seen in the microscope, which means they are present but they are so thin, which means the alveolar epithelium membrane and alveolar capillary endothelial membrane, they are attached literally closely, which means there is very less interstitium, which is normally we cannot appreciate in the even in the microscope. If you see interstitium, then there is pathology. If you see widening of the interstitium, then there is pathology. Okay, so this is how the gas exchange happens in this area. Okay, so this is very important, you should know this structures. Okay, now let us give some example. If you see lot of fluid here in the interstitium, and that fluid is going to the alveoli, it is full of fluid, nothing more than that, and this capillary is full of blood, now full of congested with blood. This capillary is, what you will suspect? You have to immediately suspect it is a left heart failure, cardiac failure. Okay, why? Because left heart failure, so in the left heart failure the blood cannot come from pulmonary vein into the left atrium, so then the pulmonary vein goes for congestion and the pressure increases, and the pressure increases in the capillary, so the fluid goes out into the interstitium and the fluid can go into the alveoli. That's why the patient gets frothy sputum. Understand? So this is pulmonary edema. Okay, so instead of fluid you are seeing proteins also, and you are seeing lot of cells coming out. That cells are all neutrophils, and you see the cells coming out, they are neutrophils, and they are destroying this alveolar epithelium, and they are also destroying this endothelium, which means some acute inflammation has happened, something triggered the acute inflammation. This fluid is having this kind of fluid is having protein as well as cells, so it is a exudate, it is a inflammation. So this is also edema, but this edema fluid has exudate, so it is a non-cardiogenic pulmonary edema. It's a non-cardiogenic pulmonary edema. Okay, I give another example. Okay, instead of you are seeing this fluids, exudate, all those stuffs, instead of you are seeing a huge granuloma, here in the interstitium. Okay, so what you will suspect? You have to suspect granulomatous disease of the lung. So likewise, you will if you know the normal, you can easily understand the pathology. So that's why I am explaining lot of basics before going to the lecture. Okay, so this is the basics part 1. Inshallah we will see the lectures. If it is needed, then we will discuss some basics.